will be recording this meeting, this webinar, and I will be actually muting everybody's line. So I would like to let you know for now that you won't be able to have any communication. If you do need to make a message, you can use the chat area in the bottom right-hand side, but we would like you to hold them until the end, when I will unmute everybody's telephone so that we can then start a discussion. So we'll just give everybody just a few more moments to come in onto the webinar.
To unmute your line, press #6.
Ollie, how are things with you?
I'm ready whenever you are.
You're still ready to go. Great.
Okay.
All right. Well, I believe we have 5 minutes past the hour. So I will hand over to Ollie. And first of all, just like to thank him very, very much, the ProteOn product line here from Bio-Rad. Ollie is a senior researcher at the CSIRO in Australia. And Ollie has previously presented at a number of conferences on this topic of antibody screening using SPR, most recently at the Lawn Conference in Australia in February. So with that introduction over, Ollie, I will pass over to you. Thank you very much.
Okay. Thank you, Laura. Thanks for giving me an opportunity to speak. This is certainly something totally new to me, but let's hope I get through it okay. So, as Laura indicated, I have spoken about this at the sort of specialized workshops at a couple of conferences, and one of Bio-Rad representatives has asked me if I then could possibly speak in one of these webinars, and that's how we got together with Laura and organized this talk. So, if we can get this going, yes. So this is, just a quick title. So I'll be talking about essentially a kinetic screening of antibody fragment libraries using the ProteOn instrument. I should add that some of these assays that are developed was also developed, or initially was developed on a Biaco instrument, so there'll be some initial experiments that were done in Biaco. So in this talk, I would like to sort of address these 5 points. First of all, I will introduce our facility, so our laboratory at CSIRO. Then secondly, Laura has asked me to do a brief overview of the SPR technology and how it is used to measure the biomolecular interactions. So hopefully it's not going to be too boring for some of you experts. And thirdly, I will then talk about ProteOn and how we use the ProteOn to measure the interactions. Fourth, then I will talk about the assay itself that we have developed over here at CSIRO. And lastly, I will just share one of the sort of anecdotal experience or an issue that I have observed using ProteOn, and it's just going to be a few slides on that, discussing how we overcome that issue. And so hopefully the talk's not going to be too long, and then I will not bore you with too much details. Okay, so CSIRO, what it stands for is the Commonwealth Scientific and Industrial Research Organization. In Australia, of course, it's a national research agency that gets federal funding. CSIRO covers wide areas of research that includes manufacturing, agriculture, biotechnology, and you can just read what I've put up there, and there's a lot more than that. CSIRO's research is performed by the 15 divisions at the moment, and we tend to partner both academia and industry, as it in a way says in the title. Our division is Division of Molecular and Health Technology. We are located in Melbourne, Sydney, and Adelaide. We have diverse capabilities that can be applied to complex problems in health and material science. And our location, or my personal location, is in Parkville in Melbourne. The building that I'm in has been here for over 50 years, and we have always specialized in protein structure, function, and relationship. And we have a strong background in protein engineering and protein crystallography. No, Biomolecular Interaction Facility in our building. I mean, I only started calling that recently, but essentially we've had SPR instruments here since 1994. That was good old BRCO 1000. More recently, we acquired BRCO T100, and very, very recently, about a year ago, we acquired the ProteOn XPR36. We are responsible for developing and performing SPR interaction assays for internal, external clients. So we support internal projects, but we also do fee-for-service contracts for the external parties, and we also serve sort of bigger projects that may require our facility. We no longer use BRCO 1000. That we put that to pasture about 2 years ago. So at the moment, we've got these two instruments, BRCO T100 and ProteOn. As most of you will be aware, there is a plethora of instruments, SPR instruments, and I'm not going to go through it, but there's just many of those. BRCO tend to be dominant, but recently we started getting more and more instruments that are very, very good competition to them, and ProteOn is certainly one of the very, very good competitors. But the way, again, I'm not going to go into it, but you can address biomolecular interaction in various ways, where you're interrogating, let's say, one protein with, one analyte, and you can interrogate 4 targets. You can, with the other instruments, you can interrogate with one analyte, you look at 400 targets. You can do 4 on 4, 1 on 16. With ProteOn, and I will be talking about that a lot more, you can do up to 6 on 6, and other instruments, 8 on 16, and so on. As I said, Laura has asked me to talk a little bit about the underlying SPR technology, and I sort of kind of explaining in a context how it works in a ProteOn. So let's see if I can do a reasonable job. So SPR phenomenon has been discovered as early, as probably late 18th century, but it's been adapted into these instruments probably in the late 80s, early 90s. The concept is that if you shine a polarized light onto a gold chip in such an angle that you get a total internal reflection, some of the photons that are hitting the gold chip will not be totally reflected, and in fact they turn into these plasmons that propagate along the surface of the gold. And as a result, some of these photons are no longer reflected, and you get this dip in intensity, or I like to call it a shadow, this sort of a line here, that's referred to as SPR, or surface plasmon resonance. Now, in these instruments, on the other side of the gold layer, you've got various matrices that you can use. In ProteOn's case, you've got alginate that you can use for attachment of, let's say, a protein onto the other side of the gold layer. And you can do that using a covalent coupling, and again, I'll talk about it a little bit more later. So what affects the SPR signal? Well, if you immobilize a protein on the other side of the gold surface, or if you bind something to that surface, you get a change in SPR signal. And more you immobilize, the more you add onto the other side, you get a change in SPR signal. So what happens? If you immobilize the protein onto this other side, you end up shifting the SPR slightly. And you can monitor that by having the detector. You can monitor that shifting signal, or shifting SPR. If you bind something to your immobilizer, in this case I've got a picture of an antibody. If you bind an antigen to it, again, you can shift that. And the beauty about these instruments is that as you immobilize, as you bind your analyte, you can monitor that in a real time. So that's different to something like ELISA, where you only monitor the endpoint of the reaction. So this is a real-time monitoring of biomolecular interaction. Other things that affect the SPR signal is the buffer that flows through on the other, or in the fluidic channel that's located on the other side of the gold layer, and of course this to deal with the issue of having a mismatch between your sample buffer and the instrument buffer. There is a requirement for referencing, and the way the referencing is done is that you've got other spot on your chip where you just pass your buffer, or that you don't have a ligand immobilized, and as a result, you can sort of just monitor the buffer response, and then you can subtract the buffer response away from your binding response. So here in this picture, you've got a flow channel that supplies the ligand or an analyte, being the two proteins or something like that, and you can sort of monitor that interaction by shifting this signal. One other thing that affects the SPR signal is temperature, but for our purposes, it's not an issue because these instruments tend to be very highly controlled, so they will measure the interaction at the set temperature, and if then you want to change the temperature, you tell the instrument to heat up to, let's say, up to 40 degrees, and you can again measure the signal at a constant temperature, and therefore the signal, in a way, in that case, is not affected by temperature because it's very, very cleverly controlled. So now the cornerstone of all instruments, and especially in this case ProteOn, is the fluidics and the cleverness of the fluidics. And I'll just address the fluidics of the ProteOn, how it works. So in here, you've got, in ProteOn, you can have up to 6 channels on your chip, and you can immobilize up to 6 different ligands using a classic NHS EDC chemistry. So you could immobilize 6 different ones on 6 ligands at the same concentration. At the sort of different concentration. You can use different chemistry, but most of the time people tend to use the amine coupling. That is, the amine of the protein is covalently attached to the chip surface. And it's a permanent covalent attachment, and that's quite important. Then you rotate your fluidics in such a way that you turn it in 90 degrees, and you can then address each of the up to 36 spots. That is, this shows the interaction, how it operates. So in this case, it shows injection of your analyte at 6 different concentrations, ranging from 100 nanomolar to 0.37 nanomolar. And each of these spots here, so in this case it shows the 6 spots, will produce a binding pair for you. So that if you inject at a different concentration, so the higher concentration gives you a higher signal, and low concentration gives you a low signal, and so on. So you can have up to 36 different binding points or binding interactions. So the beauty of the instrument that you can analyze a lot of ligands and potentially analyze at the same time. You get this sort of one-shot kinetic concept where you can inject up to 6 different concentrations in this case, and potentially no regeneration is required. Okay, this is some of the data that we have generated using in our lab using ProteOn, and in this case, what we've done, we have immobilized peptides on one of the channels. So in this case, we're just looking at one peptide and up to 4 different injections of different antibodies. So we've injected essentially different antibody fragments that have mutations in them, and you can sort of see that you're getting different kinetic profiles I should have pointed out that what you're monitoring is the association phase and the dissociation phase in this instrument. So you can sort of see that some of these, especially dissociations, are quite different from one antibody to the other. And in this case, the binding is very weak. You're barely getting any signal. I should mention something about the kinetics and how you can derive binding parameters using kinetic approach. So if you think of it that you've got a ligand and an analyte and they bind to each other to form a product AB, so you've got A plus B giving you a product AB, and it's controlled by two rate parameters. It's the association rate constant Ka and dissociation rate constant Kd. So the association rate parameter describes the collision frequency and the attraction between the two molecules, whereas the dissociation rate parameter tells you something about the stability of the complex AB. So you can then go ahead and look into this a little bit more and try to analyze what happens, and this becomes quite a bit of sort of a mathematical approach. But at the end of the day, if you buy instruments like ProteOn or Biac or anything like that, it comes out with a software that will look at your binding kinetics or binding pairs that I'm pointing out here, and they'll be able to dissect it and assign a mathematical, most of the time it's a one-to-one binding model, and derive these parameters for you. That is, it will derive the association rate constant, dissociation rate constant, by basically feeding the model to the experimental data. And it will also derive this Rmax value, which is basically a binding capacity of the protein immobilized on the chip. And just, sorry, the wrong way. And just one last thing to mention, that what does this all mean? What does these parameters really tell you? Association, dissociation rate parameters can be basically used to analyze your equilibrium. And the basic affinity, affinity can be calculated as a coefficient of the association rate parameter and the dissociation rate parameter. So basically, you generate the experimental data, then you then feed a mathematical model to them, and that will derive little Ka, little Kd, and dissociation equilibrium constant Kd. So just to point out what this means is that you've got in black you've got your experimental data, and in orange you've got your one-to-one binding fit, between the analyte and the ligand. So okay, so that's enough for the introduction to the instrument and to SPR. Now I'd like to talk about the assay that we have developed to screen the antibody fragments, and a key issue here is the key word here is the antibody fragments. Capture assays. I mean, capture assays, people have used capture assays in these instruments for 15 years and have developed them mainly for the monoclonal antibodies. And when I mean capture, some of you who may have done ELISAs, it's the same concept. Rather than immobilizing your antibody of interest directly, you capture them onto a suitable capturing agent. In this case, I've got a sort of a cartoon showing how the capture works in SPR instruments. So what we have done here, or what I've got here, is that we've immobilized polyclonal rabbit anti-mouse antibodies using covalent couplings onto the chip. And the next thing, in the instrument, we flow over and monoclonal antibodies or monoclonal antibody of interest, that being generated by hybridoma or any other technology that's around. And you capture them by, most of the time, it will be captured by the Fc region of an antibody, sort of a region that's away from the binding side of the antibody. in the next injection, we inject an antigen and monitor the binding. So the binding can, the binding curves are kind of shown here. So the first step here is the capture of the antibody. And then next, you capture your antigen. And in the last step, if you want to then reuse the chip, what you do, you strip the antibody at this level between the polyclonal and the monoclonal. So you just sort of recreate your chip like that. And for that, you can use a low pH glycine that will disrupt that polyclonal antibody versus Fc region of the monoclonal antibody. So they'll strip everything away, and you've got a chip that you can reuse. So this is just to show you again. You capture your antibody, you add your antigen, and you monitor all that in real time. And that's the key issue of SPR, that you can see everything is happening. You see everything as it's happening, and at the end you regenerate back to the back to your polyclonal level. And this chip then can be used over and over again. I think that's the key, because if you've got a lot of antibodies to analyze, or if you've got if you don't have that much money and you want to use the chip over again rather than having to pay for a new one, this sort of concept is ideal. Now, this is raw data that you generate in BioCore. So again, it sort of shows you the capture of the antibody. Here you inject your antigen, and it sort of, then you allow the antigen to dissociate. You inject your regeneration solution to strip it back to the baseline of your polyclonal antibodies. And in this case, it's got maybe about 10 different cycles repeated over and over again. So this is a basic capture of monoclonal antibodies, and that's been around for 15 years. People have been using this for a long time very successfully. I've done the same thing in ProteOn. Here I'm just showing the final data. It doesn't show the capture, but you can capture up to 6 different monoclonals. In this case, I had a protein G surface, captured it, and then following the rotation of Fluidics, I've added the antigen. In this case, it was IGF-2, and it will generate binding data, full kinetic binding data for up to 6 different monoclonal antibodies at one time. So, and this sort of shows the power of ProteOn, where you can generate this data very, very quickly, and you get a full set of kinetics. So the speed compared to other instruments is great. So capture system, what they allow you to do is they allow for oriented homogeneous immobilization of proteins by capturing through Fc. So therefore unobstructing the binding sites, and it's sort of oriented in such a way that it's readily accessible for the antigen. One of the issues is that you need to generate a stable baseline. So once you capture your monoclonal, you need to have a very stable baseline. If you don't have the stable baseline, and I will talk about this a little bit later, then you will end up having a problem of the accuracy of your analysis. The other aspect is, the surface, let's say polyclonal, has to be quite robust. So when you keep on regenerating with low pH acid or alkali or something like that, you need to be able to do this over and over again, so that you prolong the life of your chip, and you'll prolong the life of your assay if you wish as well. Now, this is very important aspect as well, that the capture should work from crude impure samples. So this concept that you can capture, so you don't need a pure protein. You can actually take, let's say, your hybridoma culture supernatant containing antibody, or in our case, we showed this with E. coli lysate, and you inject a relatively crude sample, and you achieve essentially an on-the-chip purification. And the other aspect here is, of course, you mustn't, of course, be injecting samples that will fall off your surface. So it's a bit of a sort of trial and error type of thing. If, for example, if your if your sample is too dirty, sometimes could be an issue with respect to your capturing agent, but also a chip surface. And I will talk about it, how we address that in our case. And of course, you really want to be able to, when you capture, you want to get the same results, the same data, same binding measurements, either when using pure protein or crude samples. And as I said, there's lots of papers out there that people have done it with the monoclonals, and Biakol have published on it. David Mishka at the University of Utah have published quite a few papers on how to essentially analyze that data, and there are many other publications. Well, we could do this capture of the full antibody. So this shows the full antibody here. You've got the binding site at the top, and you've got the Fc region at the bottom. And most of these capture assays with monoclonal is through the Fc region. In our laboratory over the years, maybe not so much recently, but we have always been expressing antibody fragments, either FAB fragments, single-chain Fe fragments, or V-domain fragments. And these can be readily cloned into E. coli. They can be readily displayed on a phage surface. And people have created, and we have created libraries of these fragments with various mutations in them. And one of our issues was that how quickly we can screen potential candidates, better binders, or when we introduce some specific mutations. How can we screen them very quickly? The conventional way would have been that we express these, and then we would have used the conventional protein purification methods to purify these. We would then take their corresponding antigen, immobilize the corresponding antigen onto the chip surface, and set up a kinetic analysis by injecting the antibody over the immobilized antigen. That process is quite long, and no matter what assay you're using to pre-screen these fragments so you can narrow down, it takes a long time to get a full kinetic analysis of your candidate binders. And it takes a long time. So we were always keen to develop some sort of assay where we could very easily analyze our candidate binders. So most of the time when we express these proteins and antibody fragments, we would have a tag at the C-terminus of these fragments. Most of the time it would be a hit tag, or we would use a flag tag. And we, in the past, have tried to capture these antibody fragments onto the chip surface. So in this case, we had a anti-hit tag antibodies on the surface. We then injected our protein, captured it on the surface. But what we always noticed with some of these tags, that they were coming off the surface very quickly. And if you get a chip like that, it's pointless trying to inject your antigen and hoping that you end up getting a good, accurate kinetic analysis. The problem here is that it decays just too fast to be able to sort of reproduce this and being able to generate a good subtracted data that can be used to analyze a difference in binding that's only twofold, in affinity. So this baseline is just not good enough for that. I'd say about 5 years ago, we came across this interesting system, and it's based on two proteins binding to each very tightly, binding to each other very tightly. I'll just tell you something about the background of these two proteins. So, protein we're talking about here is the DNA E7 protein and its binding partner immunity 7 protein. Now, DNA E7 protein is essentially a subdomain of a colicin, in this case E7 or protein that's produced by bacteria as part of their stress response. So if bacteria is competing with another bacteria, it will produce this colicin E7, in this case this E7 protein, that will find its way into the competing E. coli, and through this catalytic DNA E7 subdomain will chew up the competing E. coli DNA. However, this DNase E7 is fairly non-specific, so in order to prevent the self-digestion, so the bacteria produces it, in order to prevent this self-digestion, the bacteria will at the same time produce the immunity 7 protein that will bind to this colicin E7 and block the action of the DNase E7. And it's secreted as a complex, and when it gets inside the competing bacteria, it's somehow dissociated. That process is not very well understood. Nevertheless, the reaction between the immunity protein and the colicin E7 is really, really tight, and it's been measured using various methods, not SPR, but other methods, that it's in the order of 10 to the negative 14 molar kb. So that's sort of approaching the streptavidin binding type of interaction. So when we were using this system in our lab for something else, and I sort of thought that might be an interesting way of being able to immobilize our antibody fragments onto the chip surface. So the idea was to make the DNase E7 protein, couple it covalently to the chip surface, and at the same time make a single chain or antibody fragment fused with immunity proteins at the C-terminus. So that's the immunity protein in green, and that's the single chain Fv fragment. That will then, this chimera will then bind to the DNase E7 protein on the chip, and will bind very tightly, allowing for then subsequent injection of antigen and measuring the kinetic, or measuring binding or kinetic, doing kinetic analysis between the antigen and antibody. So for that, we first had to make the DNase E7. So this slide just shows the method, how it's made, and this is well documented in the literature. So all we did, we just sort of got it out of the literature, made it, and it worked really well. The only thing to mention here is that if one wants to produce DNase E7, because in E. coli it's so toxic, it has to be made as a sort of it has to be co-expressed with the immunity protein. So what you're actually doing, you make a complex, your expression produces a complex of the DNase 7 and IM7 that can then be purified. In this case, we had a histec at the N-terminus of the DNase E7. We purify the complex and then use the ion exchange chromatography and other methods to separate the two proteins. And by doing that at a low pH, we were able to obtain pure DNase E7, and as a byproduct of this expression is, of course, immunity 7 proteins. But in this case, we didn't really need it. We were producing DNase E7 because we needed to immobilize it onto the surface. Now, we have, as I said, we've expressed a number of different formats of antibodies in the lab. So, of course, we wanted to introduce the immunity protein, or make the immunity protein conjugate with single chain. We've done it with a diabody. The diabody is just a single chain with a shorter linker. We've done it with the shark domain, with a single domain or single pH domain. And we've used the immunity protein to SvFv fragment as well. So you've just got a cartoon here of different formats that we have expressed and analyzed using this capture assay. So for the rest of my talk, I will just home in on examples how we've done it with the single chain and how we've done it with the V domain. Separately, we've published most of this work in a publication which I will show right at the end, and that also describes the diabody, how it works for the diabody, and how it works for the SvFv fragment. So, okay, again, this was developed before we had access to ProteOn, so this shows the BIA core data. Now, we'll go through this fairly slowly because it's quite a complicated slide. So, there is 40 overlapping cycles shown here. So each cycle consists of injection or capture of recombinant antibody fragment onto the DNase E7 coated chip. So that's in this part shown here. And then injection of either buffer only, so it's just injecting a buffer that's in blue, or injecting an antigen. In this case, it's a 3-2G12 antigen, which is an anti-urea antibody to the single chain that you've got here. So that shows binding here. And in the third step, it shows the regeneration of the surface back to the DNase E7 level. So as I said, there is 40 captures, 20 injections of buffer only, and 20 injections of recombinant antigen. Here in the inset shows that basically monitors the capture. How many, each cycle we captured roughly 340 RU of the fusion of antibody IM7 fusion. So what I'm trying to show here is that the baseline is very reproducible. So you see very nice baseline, no fast dissociation, very reproducible. Same thing when we're injecting the antigen, very, very reproducible. The capture itself, the difference between here and the last one was about 2%. Now, for comparison purposes, I also had this one cycle, which was essentially an instrumental error. That's this cycle here. And it was not actually an issue with the capture. It was just an issue with the SPR signal. So what this slide shows is shows the really good baseline, or post-capture baseline, and shows a very, very good reproducibility of this assay. We then went ahead and measured the interaction between in this case was Antigen SvFv IM7 captured and how and binding to 3,2-G12 Fab. We've done it with the pure sample of this single chain conjugate and with the sample that was just in an E. coli supernatant. And here just shows the corresponding kinetic data and kinetic fits, and the data that we are getting were highly, highly comparable. So we were really pleased with it. Well, at this point, what happened? I found out about the ProteOn instrument, and I spoke to Bio-Rad Australia and eventually Bio-Rad US, and they offered to analyze some of my capture system over in the United States. And later on in that year, which was about 2 years ago, I was able to test the instrument by having it on loan. So that's back in 2007. So the data I show you is showing the performance of this assay that I just described in the ProteOn instrument. So just to remind you how it works in the ProteOn instrument. So the chip contains immobilized, or we immobilize the DNase E7 into each of these 6 channels in this sort of ligand direction, and then secondly we capture our antibody IM7 conjugate in 5 channels with the 6, and then we're leaving the 6th channel blank, so we just inject the buffer. So doing it this way, we can analyze 5 different antibody conjugates at the same time. We then rotate the fluidics, and inject the analyte, and again we monitor the association, and then by washing we can monitor, in replacing the analyte with the buffer, we can monitor the dissociation phase. So as I said, you can analyze up to 5. Doing it this way, we were analyzing 5 clones at the same time. So you've already seen this. I'll just move on. So this just shows the data. I think this is essentially my data once I got the instrument here in Australia. Just showing the same thing that I've showed with my with our vehicle instrument. That is, we capture the Antigen SvFv IM7 in 5 different channels to a certain degree at different densities, and we injected the free 2G12 and generated very nice data showing the binding and showing good sort of comparable kinetics. So that was just a proof of principle. Next, we wanted to analyze the V-domain library that Stuart Nuttall had here in the lab, and he generated this library and essentially Affinity matured this library using phage display. And we were keen to look at their kinetic profile, and we were keen to do it in such a way that we don't need to purify these clones. So the whole idea was to take the phage clones, clone them into bacteria, and then grow them in this case we took 48 clones. We basically picked the colonies, grew them in these 96-well plates. They are 1 96-well plates. So the concept of expression here, I should have said that before, but the idea is that these antibody fragments are expressed into bacteria. The fact that after a while you get a leakage into cultured supernatant. So if you express overnight, generally you've got quite a bit of your antibody fragment in E. coli. And the only thing that we started doing here at this point, that we started filtering our cultured supernatants to remove material from our samples that were affecting our SPR. So we use these supernatants sequentially for a 0.22 micron plate and then for the 100 kilodalton plate. By doing that, we were semi-purifying our samples. And theoretically, anything that's bigger than 100 kilodalton that is just to improve the performance of our instrument and also protect the fluidics of the instrument. So this is the cartoon of the shark VH domain that we were 5 channels, and as I said, we left the 6th channel for blank purposes. Following a rotation, we injected the antigen, that being malaria. So that way we were able to analyze 48 clones, and I did it in 8 hours, but at the time I wasn't very skillful with the instrument. I think now that could be brought down to probably about 4 hours. So this is the raw data generated from this experiment, and as you can ranging from no response to very good response. Now, the no response, we subsequently found out that these clones were either non-expressing at all because we just randomly picked a colony and we found that clone didn't express. And the reason for no expression was that, which is a common thing in phage display, sometimes you can get a mutation that will truncate your protein expression. Therefore, we could, of course, never capture any of this material onto the chip, and subsequently we didn't get any response when we injected the antigen. So out of 48 clones, I think only about 39 expressed and gave us useful kinetic profiles. And I eventually was able to group these into, I guess, 7 different groups based on their kinetic profiles. The way we did that was using this kinetic distribution plot. And all you're really doing, you take your determined dissociation rate constant KD, you take an association rate Ka, and plot them against each other. And these dotted line represent the affinity. So this is so that represents a 1 micromolar affinity, that's 100 nanomolar affinity, 10 nanomolar affinity. We knew that our clone was starting clone before it was affinity mature was in the order of about 200 to 300 nanomolar. So we kind of presumed that all these clones were more or less the wild-type clones that didn't have any significant mutation in them that improved the binding to the antigen. But then when we started looking at some of these clones that kinetic profiles of these clones, we could easily group them based on their where they sort of fit onto this kinetic distribution plot. Now, I have subsequently sequenced each of these clones and was able to possibly elucidate the affinity or the reason why these clones fit into let's say group 1 or group 2. So, for example, group 1 had 2 significant mutations in the sequence of the VH domain. Both of them were located in either CDR1 and CDR3, that is the regions that recognize the antigen. And it was the proline to leucine mutation and alanine to threonine mutation. Group 2 had only that proline to leucine mutation, didn't have the alanine to threonine mutation. It has some other mutations, but obviously these mutations are located in regions that didn't make that much effect towards binding. And so on. We were able to lots of interesting groups. Groups 3 and 4 were quite interesting. Group 3 was essentially just one clone. Was the proline to serine mutation. So if you just look at it, it's here. So dissociation rate constant is slightly faster compared to some of the when you got a leucine in that position. Group 4, it had a leucine mutation, but some other mutations in them that affected the binding, and again, that mainly affected the dissociation rate constant. Group 5 had a mutation, or was it aspartate to glycine? That didn't make that much difference, but there was some difference to it, compared to wild type. Interesting mutation was group 6. It was located. The mutation was located outside of the CDR3, but just outside, in this alanine to threonine mutation. And if you look at your kinetic profile, that mutation doesn't seem to be affecting the dissociation rate constant as much. It's equivalent to wild type, but it's affecting the association rate constant. And I think the difference is about twofold. So we were extremely pleased with this result because it could pick up very subtle differences in our kinetic profiles. So this assay was able to distinguish, and the assay was done with the crude samples. The differences are minor, but are significant in terms of these mutations. So, and being able to use ProteOn, we were able to do this really, really fast, being able to obtain full kinetic data for each clone with every injection, and therefore be fairly confident in, with our results. So again, it sort of shows the kinetic profiles just to sort of highlight them. Group 1 dissociation rate constant is much slower than, for example, when you look at the group 4. So just to summarize this, this work, DNase E7 immunity protein 7 interaction is very well suited to capture various antibody fragments onto the DNase E7 sensor chip surface. We have also found that by adding the immunity 7 protein onto the back of our antibody fragment, didn't significantly affect the expression and folding of our antibody fragment. So that's important. You don't want to add a fusion partner that will destroy your expression. And the fusion partner is relatively big. It's 10 kilodaltons adding onto an antibody fragment that ranges from 14 to 50 kilodaltons. So it's a significant chunk of that's being added. Now, importantly, this capture system generates stable baseline that allows for accurate and kinetic binding analysis of antibody fragments. So we were able to pick up twofold differences, and we were very confident in those that there is a twofold difference, and that twofold difference we were able to associate with a mutation. Now, the other important aspect is that the DNase-E7 surface, that is the protein sitting on the chip surface, is so robust that it allows you to do repeated analysis. And let's say in Biaco, we have regenerated 200 times or more, and the surface was still performing very well. Now the capture works very well with the relatively impure E. coli samples. And we were able to demonstrate that this capture system works very well in Biaco and extremely well in ProteOn when it comes to screening. Okay, just to sort of acknowledge people that have done this work, just put up our publication that was published earlier this year, and people to highlight is Ralph, who has started all this work before he left us and moved to Switzerland. Megan and Leslie, that they've done a lot of the work, preparing proteins, helping out with setting up, and Megan doing the assays. Stuart Nuttall and Louise Duffel helping out with the shark libraries, and Lee developing a protocol of purifying the DNA C7. Okay, I just have I have sort of finished the main part of the talk, and I just have about 4 slides addressing some of the issue that one can see with parallel biosensors, and in this particular case, my experience with the ProteOn. And it relates to capture of your ligand onto the chip surface. And for whatever reason, sometimes it's through either my fault or operator's fault, or sometimes the way you setting up your immobilization, you can end up with sort of an inconsistent immobilization or sort of a gradient immobilization across your ligand channel. And the reasons for that could be just the way you're setting up your method, or sometimes you need to do sort of like an experimental design, that is, you have to experiment with your protein concentration or how long you inject for. I have found that some of these optimizations can be a long process, and this is just this just shows what can happen. Sometimes in this case, we injected the biotinylated peptide onto a streptavidin surface. So we made a streptavidin GLM, we immobilized streptavidin on a GLM chip, and we found that each of these spots had a relatively different amount of protein immobilized. And since then, I've done this maybe 6 months ago. Since then, I started learning about how to do this a little bit better. But I guess the point I'm trying to make here is that this will happen, and it can happen for whatever reason. And how do you deal with it? Do you go and try to mobilize another channel, or do you not worry about that? And I've got this method, or we've developed this method that it's fairly straightforward, but allows you to deal with that issue fairly quickly. Okay, so what you've got here is a differentially immobilized ligand. That is, therefore, if you want to do a bunch of kinetics, you cannot really relate each of these spots to having the same Rmax, same maximal binding capacity. Therefore, you have to do something different. Now, this just shows that we've taken a distinct chain against this peptide that was immobilized on a chip and injected it at 6 different concentrations, ranging from 64 to 0. And when we tried to fit it with one-to-one binding model that had where we had a global, where we fit the global Ka, Kd, and Rmax, we were seeing a poor fit. And of course, we would see a poor fit because the Rmax isn't the same; it's different. So one way to go around it is basically that you fit it with the global Kn and Kd, that is the Kd constraint for all the curves, and you fit it with each curve with a local Rmax. And this sort of shows you your result. So your software will predict that your Rmax is different, so it follows what we saw when we immobilized, that is, one spot's got higher maximal binding capacity than the next, and so on. Nevertheless, the binding that you get is fairly believable. The fit's quite good. It's not strictly speaking ideal if you do it this way, because there could be other reasons why you're not getting a good fit. But because you know that your Rmax isn't the same, it's fair enough to constrain only 2 parameters and leave the third parameter local, and therefore you can be fairly confident that your answer, your measured answer in terms of Kn and Kd is accurate. Well, there's one other way to deal with this to confirm that this is acceptable. And rather than doing a single one-shot injection, you do 6 of them. And all you're doing that you address each of these spots from A1 to A6, you address them separately. That is, in the first injection, you've got your 64 nanomolar, 32 nanomolar concentration like this, and so on. In the second injection, you vary it. So after 6 injections, you would have injected each of these concentrations over a single spot. And in a way, to a degree, it's like the old instruments, like the echo instrument and so on, where each of these concentration has addressed a single spot. Point to make here that in order to be able to do this assay, you have to be able to regenerate. So each time you do injection, you regenerate and so on. So you need to verify your regeneration conditions, which you may not need if you just do one-shot kinetics, as I have outlined before. So this way, you're going to end up with 6 sensograms like here. And in this case, you can then fit them with the global Ka, Kd, and Rmax. So all 3 parameters are constrained for each of these curves. And then you determine your binding. And at the end of the day, what we found that the binding, either using that single path and a local Rmax, the binding parameters determined, they're virtually identical. Just to show you, so the values here vary in the order of approaching 2 to the power of 5 and 1.2 times 10 to the negative 3 using the local, local Rmax. And in this case, it's around about 1.9. Well, on average 1.8. That is for 10 to the 5, and still Kd dissociation rate is about the same. So either way you do it, this is one way how you can troubleshoot if you end up with a differentially immobilized channel. Right, and that's it. So that's thanks for your attention, and I'm happy to take questions.
Thank you very much, Ollie. That was great, and thanks ever so much for the little troubleshooting tips there at the end. I've taken everybody off the mute, so if anyone has any questions, please feel free. Ollie's ready and waiting. Do we have any questions? Okay, I have a question. So Ollie, with your short antibody fragments, what's your next step? Do you go ahead and use those as they are in kind of clinical or in animal models, or do you make them into full length?
So in this case, some of these what you would do next is either rebuild a whole antibody or set them up for your clinical trials or something like that. So the method that I'm demonstrating here is just purely screening method. You may want to use the short chain fragment or VNAR in your biological assay. It just sort of depends where you are. In our case, most of these were just a sort of a proof of principles and how they fit, like in case of those shark domains. He was really interested to be able to affinity mature some of these and to find out how they bind to AMA1 proteins. So some of these proteins were then taken and made as the V domain on its own and put into crystallization trials. And he wanted to understand how they bind to the target protein. So he was really trying to understand the protein-protein interaction at the sort of a crystallography level.
Oh, that's really interesting. So, and I just had another question about the assay development. How hard is it for you to decide on which is the best capture? Because you talked about capture earlier on. I wondered if you had any guidelines that that might help people who are perhaps not using your specific capture method, but perhaps using protein A or an anti-IgG or an anti-Fc.
Look, I guess everybody's got their favorite one. I mean, for a while I quite like the rabbit anti-mouse antibody. Were really good against monoclonal, but as soon as we started dealing with human antibodies, I either had to use goat anti-human, but lately I've been using protein G quite a bit and with relative success. One of the issues with protein G is that it doesn't immobilize very well on any chip because it's it has very low isoelectric point, And if you're using direct amine coupling, that can be an issue. Nevertheless, we've got a reasonable method where we raise the temperature and we can get a reasonable amount of protein G onto the chip. Because the whole idea is if you want to have an efficient capture assay, you've got to be able to immobilize quite an amount of your capture agent if you wish. And therefore then have a great capacity on your chip to be able to capture and retain. Because some of these captures are not just being able to capture, but no matter what people think, there's always some dissociation going on. So more protein that you capture, you can then rely on what you refer to as rebinding or mass transport effect. As the molecule dissociates from one, let's say, protein G molecule, there is a 1 nR nearby that will anchor it. So you essentially see a nice baseline that doesn't drift.
That's great.
I've got a question.
Thank you. Go
from
ahead.
Jason. Olan, I was just wondering your feel for screening a higher affinity interaction, because you were looking at around about 200 nanomolar there, and how the ProteOn holds up with, say, sub-nanomolar binders.
Okay. We did have, in that, when I showed a quick cartoon of the monoclonals against IGF-2, we had some binders in there that were in the order of certainly sub-nanomolar. We were able to measure it fairly well, but of course, issue is if you get a bit of drift with any of these things, then at let's say 100 picomolar and lower, that drift may start making quite a bit of difference to your accuracy, if you wish. Of course, one of the things to do is to do it at high temperature. I have successfully used ProteOn at high temperature, but not necessarily with the monoclonals. I have done it with the IM7, DNAC7, and in that case, I had binders in the order of about 500 picomolar, and that it worked okay. It certainly did work.
How high a temperature were you going up to with your DNA system?
I think 35 degrees was my highest temperature.
Okay.
So that was my highest. I was able to get a fairly good data. Of course, at higher temperature, any instrument starts to struggle no matter what. Your data don't look as good, but in the overall picture, it's okay because it's still a screen. Yeah, I would say yes. I mean, it works quite well. I guess you can come and see me here, and I'll show you the data.
Yeah, sure.
You're lucky. So I did actually have a question from Ki Jong Hong, and he was asking about can we modify this for use with oligonucleotides?
Sure, yes.
Yeah. And
yes, so, yeah, sorry, I wasn't sort of clear enough at the start. I guess my experience most of the time are proteins, but of course with oligonucleotides, one of the classic ways to do that is to mobilize streptavidin or neutravidin, or you can from Bio-Rad you can actually purchase a neutravidin chip that's ready made for you, and then you take your nucleotide that's biotinylated and you can immobilize your nucleotide onto the neutravidin surface. So yeah, it works really well, and then you can interrogate your nucleotide with a protein, or you can interrogate it with another nucleotide. The only issue is how is the size of your molecule and how sensitive your event can be.
So thank you, Ollie. There any other questions there?
I was wondering if Ollie would like to comment on non-specific binding with the alginate chips.
Non-specific binding. Look, I may have seen occasionally, but I would say it's no more than you would get with Biacor. Most of the time, non-specific binding is related to the protein that you're using, or your analyte that you're using, and the concentration that you use. It varies. I mean, I think it's a case-to-case scenario. I suppose non-specific binding, if it's minor, it doesn't matter unless you're measuring high affinity antibodies. Then it can start sort of make quite a bit of difference. Or the other end, I suppose, when you've got a really low affinity, because that can make a difference too.
That's a good point. There's another question that's come up on the chat here, and can you see that there, Ollie? It's, have you met a case where sometimes you have a clone that was positive in an ELISA test, but somehow there was no response when you've worked on the ProteOn or by SPR?
Yes, very good question. It happens. We had some Ricin antibodies and had a panel of 6 Ricin antibodies, and one of them binds very well in ELISA and doesn't bind in SPR. I have this belief, and if you read literature, people anecdotally report that. The reason for that is that most of the antibodies that people deal with have been developed or, have been screened or have been selected using ELISA play. So what happens, you immobilize your protein, you immobilize your peptide, and in the process you may denature it partially. And if you denature it, then you're selecting antibodies against the regions, or the antibodies bind to the regions that are not seen in solution, that are not seen in SPR. So you've got to appreciate whichever way you, even though, concept is similar, you mobilize on an ELISA plate or you couple it to a vehicle chip, the protein could be different. So if you select it you select your antibody against that protein on ELISA, then there is a chance that you sort of get it to a point where it won't bind in solution. And one way to check it for you, if you could set up some sort of in-solution assay. Sometimes if your affinity is high enough, if you take your antigen and antibody, put them together and pass them through a gel filtration column, you may not see binding either.
Yeah. So it depends on the method you're using.
Well, the method of creating that antibody can affect it.
Yeah. Well, that's a good point. Were there any other questions? Okay. Well, I will just wrap this up by saying a big thank you, Ollie. That was absolutely wonderful, and I learned a lot. I'm sure everybody else learned a lot, and thank you everybody for your questions. The webinar has been recorded, and I will send out the recording to everybody by email tomorrow. And if you do have any questions, feel free to email myself, and I can put you in touch with Ollie if you like. So thanks everybody for your kind attention. Ollie, any last words there?
Yeah, just if you want to email me or whatever, if you dig out that reference that I put up on a sort of near the end, my email's on that. So and feel free to email me.
Thank you. Thank you everybody. Take care and have a great day to you all.